In Chapter 21, we talk about genomes and their evolution, and specifically in 21.5, we're going to look at genomes and how genes are duplicated, rearranged, and how mutation of DNA can contribute to the evolution of genomes. We know the basis of evolution is change, and change at the genomic level happens as a result of mutations. We also know that the earliest forms of life had a minimal number of genes, so they only had genes that were necessary for their survival as well as their reproduction. Over evolutionary time, we see that the size of genomes has increased and we tend to get some duplication of genes. So instead of having one copy for a necessary gene, you would then have two copies. And as long as you keep one copy intact that is necessary for survival, it does lend itself to that second copy of the gene mutating and acting in some sort of novel way that can change your phenotype. And if you get enough of those mutation events, then that becomes a raw material for gene diversification as well as speciation to occur. So you tend to get a divergence of one species into two different species as those different mutations tend to accumulate in a genome. Duplication is one way in which we see evolution of genomes and an accumulation of differences among species. So we know that a duplicating event in a chromosome occurs as an accident in meiosis. So this can lead to one or more extra sets of chromosomes, which we consider the condition to be polyploidy. Genes in one or more of the extra sets can then diverge by accumulating mutations. So you have one functioning copy and then you have another copy that can be subject to mutations, and so it changes. And if those changes to the gene are beneficial, you tend to see those variations persisting in the organism, and that is if the organism survives its environment and survives to reproduce. Those traits then get passed along down the chain. We also know that chromosomes can have alterations to their physical structure. An example would be humans and chimpanzees. Humans have 23 chromosomes, whereas chimpanzees have an extra chromosome and they have 24 pairs. If we were to look at the divergence of humans and chimpanzees from a common ancestor, we see that the common ancestor had 24 chromosomes as well, but in the human lineage, 2 of those chromosomes fused together. So it decreased the total number from 24 to 23 chromosomes. That same event, that fusing of 2 chromosomes, did not happen in the chimpanzee line, and therefore anything that diverged from that ancestral line still has 24 chromosomes. Duplications and inversions result from mistakes during meiotic recombination. And so looking at an analysis between chromosomes of humans and 7 other mammal species can help us to paint a hypothetical chromosomal evolutionary history. So we talk about evolutionary history in terms of similarities between homologous structures, DNA sequences. We can also do that for chromosomes as well. Here's an example of a comparative analysis of chromosomes between humans and mice. If we were to look at chromosome 16 on humans, we would see 1, 2, 3, 4, 5 different sections of the chromosome. Those are blocks of DNA sequences. And looking at the mouse chromosomes, we can see the same blocks, the same 5 blocks on different chromosomes in the mouse. And what this suggests is that humans and mice shared a common ancestor, and those 5 sets of DNA, or those blocks on the chromosome, stayed together up until the point where there was a divergence between the most recent common ancestor between humans and mice. So comparison of chromosomes gives us a really good idea of when divergence occurred, and it's just another key to figuring out taxonomic classifications. So we know that the rate of duplications and inversions of chromosomes seems to have sped up starting at about 100 million years ago. This is the same time when dinosaurs went extinct and mammals diversified. So we had the age and the rise of the mammals. Chromosomal arrangements are thought to contribute to the generation of new species, in that with each chromosomal duplication or inversion or fusing, you tend to get a evolution of your genome, so your chromosomes are changing. We used to think that the breakage points where those chromosomes are swapping out and duplicating was completely random. And now we're finding that they're actually breaking off. The chromosomes are breaking at the same spots. And we call those spots where they're breaking hotspots. And we've also found that some of these chromosomal hotspots are also associated with diseases in humans, congenital disorders. So the more we study chromosomes and their evolution and how they've changed over time, the more we are starting to learn about our species as it exists today, as it existed in the past, and what that means in terms of our medical history for humans. When our meiosis machinery is not working properly, we get unequal crossing over, so meaning we're not swapping out two identical or complementary pieces of chromosomes during prophase 1. It can result in a chromosome that is missing an entire piece of a gene. And then another chromosome that has a duplication. It has two sets of genes for that particular trait. Transposable elements provide sites for this crossing over to happen between non-sister chromatids, and they're very important. On the next slide, we're going to see how they work. So here we can see we have two non-sister chromatids, so they're not the homologous pair that should be lining up, and they have what we call these transposable elements on them. So these transposable elements line up, and you have genes that are not complementary to one another, and you get crossing over at these transposable elements. The problem with this is that you have an incorrect pairing of your two homologs. So you're not swapping out complementary chromosomes. You're not swapping the same genes between two of the same chromosomes just from different parents, but you're actually swapping a gene for one particular thing with a gene of another. So you wind up with this organism that has two copies of a gene for one particular trait, and another organism that may have no copies, or another chromosome that may be missing it entirely. So this would be an example of how transposable elements lead to genomic shuffling and a rearrangement of the chromosome, as well as duplication and deletion. One of the genes that we study to look at the evolution of a genome is the Human Globin genes, and the genes encoding various globin proteins evolved from one common ancestral globin gene, which had initially duplicated and then diverged onto and was transposed onto different chromosomes about 400 to 500 million years ago. After the duplication events, you start to get differences in the genes in the globin family, which arose due to random accumulation of mutations. So remember, as long as you have one functioning gene, any duplicated gene can be subjected to mutations. And as long as they're not adverse in effect to the organism, they're going to tend to be passed down. That organism is able to survive their environment and pass that duplicated gene down. And so that's where you tend to get differences between species, as in those duplicated genes. And you would see an accumulation of those mutations, and you can travel backwards and find out where that divergence occurred and about how long ago it occurred. So here's that example we were just talking about. We started with one ancestral globin gene, and we know that there was some sort of duplication event. So that gene was duplicated, and then there were two copies of that gene. There was a mutation to both copies, and neither were disadvantageous. Both allowed the organism to survive and reproduce. And so then we referred to one as the alpha globin gene and one as the beta globin gene. And these guys were then transposed to different chromosomes. So we had non-sister chromatids engaging in crossing over events. And so the alpha globin gene was transposed onto chromosome 16, whereas the beta globin gene was transposed onto chromosome 11. And over time, we see further duplications, mutations. So we have a whole multi-gene family for both the alpha globin gene as well as the beta globin gene. So we can track these mutations back and figure out where those duplication events occurred, how long they occurred ago, and then we can compare between different organisms as well. Our modern day oxygen binding proteins are the globin from the globin genes. And we know that these gave rise as a result of duplications of genes and random mutations to give us what we now understand as our oxygen binding proteins. We can compare amino acid sequences for various globin proteins, and that does support our model for gene duplication and mutation. So looking at our alpha globins, we know that alpha globins are more similar to each other, and beta globins are more similar to each other than alpha and beta are similar to each other. So looking at those amino acid sequences, it's a way for us to classify, organize, and then trace back where those specific duplication and mutation events occurred. So how do we get these new genes with novel new functions? We know that the copies of some duplicated genes have diverged so much in evolution that the function of the proteins that they code for are now very different than what they were originally. An example of this would be the lysozyme gene that was duplicated, and one copy of that lysozyme gene evolved into the alpha-lactalbumin gene in mammals. The lysozyme gene, as it was initially, its original function was as an enzyme that helps to protect animals against bacterial infections. Once that gene was duplicated, that other copy of the lysozyme mutated into alpha-lactalbumin, which is a non-enzymatic protein that plays a role in milk production in mammals. So we start out with just a simple duplication event, and after mutations, we still have our functional original lysosome gene. But now we have this novel new gene that plays a role in milk production that has a whole new function compared to the original gene's function. Genes can also be rearranged, and the parts of the genes can also be rearranged. And we see that in exon duplication as well as exon shuffling. If you were to duplicate and reposition an exon, that would contribute to genomic evolution. Errors in meiosis can also result in exons being duplicated, so you have two copies of that expressed gene on one chromosome and then deleted from the homologous chromosome, so that copy winds up being on one and not on the other. So you have two copies on one absent from the other. In exon shuffling, you get errors in meiotic recombination. This leads to mixing and matching of exons either within a gene, so you can have exons being shuffled around, or between two genes that are non-allelic to each other, so they're not coding for the same traits. Here's an example of exon shuffling and exon duplication in the modern day gene that codes for the tissue plasminogen activator, which is a protein that controls blood clotting. We see that the gene itself is made up of several different genes. We see the fibronectin gene is in there, and the epidermal growth factor gene is also a part of it. So that came to be part of the TPA gene due to exon shuffling. And we also see plasminogen genes, which is K exon, was also put into there. That particular gene, the K gene, was duplicated. So that expressed region made a copy of itself. And so as TPA exists today, it is a result of rearrangement in exon shuffling as well as exon duplication. So transposable elements do contribute to genomic evolution. Having multiple copies of a similar transposable element can facilitate recombination or crossing over between different chromosomes. If you were to take a transposable element and insert it within a protein coding sequence, that can block protein production. So your normal transcript machinery cannot read that properly, and therefore you are not going to synthesize that protein product for that gene in question that has a transposable element. You can also insert transposable elements within a regulatory sequence, and this can increase or decrease protein production depending on where in that regulatory sequence a transposable element is. So you can see more of that protein product or less. Transposable elements also carry genes or groups of genes to new locations. They can create new sites for alternative splicing in an RNA transcript, so you wind up with a new gene product. And in all cases, these changes that are caused by transposable elements are typically detrimental to the organism, but every now and then they can prove advantageous to the organism. So they can be selected for in a particular organism in a particular environment. And so getting an accumulation of those traits that are advantageous gives rise to new species as well.